Saravana Ramasamy. I'm based at Imperial College, London. So I'm the moderator, as Caroline introduced already. So first, let me start with some housekeeping instructions. So everyone can ask questions during the talk also and after the talk. So you can post your questions in the Q&A box, and then if someone finds the question is already there and you're also interested in the same question, you can do the upvote so that I can know how many of you are interested in the same question, and that will get priority. And yes, this today's session has two sections. The first, we are going to allow Stefanie to talk and explain the topic, and then question and answer. And after that, the second session will be closed session, where PhD students will have the opportunity to interact with us. And Stefanie, you can ask all the questions like scientific or career. So we can have a closed session in that. So to introduce the topic, let me share my screen. I'll give a brief introduction. I hope you'll be able. You're seeing my screen. Sorry.
Yes.
Okay. So today's topic is communication in the aging and disease heart by Professor Stefanie Dimmeler. So just to introduce the importance of this topic, so based on World Health Federation, Heart Federation, you can see that cardiovascular diseases is the world's #1 killer. So you can see the importance of working on heart. So if you look at the number, you can see it's 18.6 million deaths every year. So that's really a huge number. So let's look at the statistics. So here you can see the cardiovascular deaths are the #1 killer, and it has surpassed cancer. So previously, a decade before, cancer was the highest killer, but now you can see cardiovascular diseases. So it has become more and more important to understand heart and the disease related to that. So this is another graph. If you look carefully, you can see that the axis here in the x-axis, you can see the ages, and this is the incidence of cardiovascular disease or deaths. So you can see the top two are for cardiovascular diseases, and the bottom two lines are for the non-cardiovascular deaths. So you can appreciate that there is also an age factor in the incidences of cardiovascular diseases. So the aged people have high incidences of cardiovascular diseases, which makes us to wonder why there is a relation between aging and also incidence of cardiovascular diseases, and why. So I will not take much time. So apart from that, the heart has many important cell types. The cardiomyocytes, which form the major cell type in heart. Apart from that, endothelial cells, fibroblasts, and macrophages form important population in the heart. So today, Professor Stefanie Dimmeler will talk about how these cells interact and how they behave in the aging and disease heart. So to briefly introduce about Professor Stefanie Dimmeler, she's Director of Institute for Cardiovascular Regeneration in Frankfurt. She is also a spokesperson of Cardiopulmonary Institute and also chair of German Center for Cardiovascular Research. So just to indicate the importance of her work, she has been one of the highly cited researchers for several years now. So that clearly you can understand how important or relevance of her findings. So apart from that, she also received many awards for her work, and also she is associated editor of many of the journals at the moment. So, so now I stop my introduction and let Stefanie talk about her findings. So, please, Stefanie.
Well, thank you very much. Let me first share the screen and see whether everything is working.
Yes, yes.
Yes. So you also see my pointer? Then
yes, I can see your
Thank you very much for the kind introduction and also for the invitation to speak today. And it's now my pleasure to bring you some insights. Part of them are really unpublished and new. How cells communicate in the heart and what is the relation to age and disease. I was also thinking about that the audience might be broad, and so I wanted to bring some basic background and echo in the end what has just been shown also introduction. The heart is a muscle which pumps blood in, and it has a lot of work to do. So every day, 100,000 times the heart beats. And this means 2.5 billion times in a normal life. And the major cell type in the heart is the cardiac muscle cells, which are the ones which are pumping blood. And there are roughly 2.2 to 3 billion cardiomyocytes in the heart of a normal human. And of course, vascular disease are the leading cause of death still. And this is in part due to heart dysfunction, because if the heart can't pump blood anymore, of course, the entire body is devoid of oxygen and cannot live. The problem with the heart is, and why it's one limitation why it's easily affected by disease, is that the heart has a poor regenerative activity. So in contrast to skin or intestine or many other organs where there is a lot of regeneration ongoing, the heart can, if at all, can only regenerate at very early stages during life. In mice, it has been nicely shown that cardiac regeneration only occurs within the first 7 days after birth. In this time, also if you induce a myocardial infarction, which means to block the blood supply or you cut a part of the heart, there is a full regeneration ongoing. In adult mice and humans, this doesn't work nicely. You can see a mouse heart or a human heart after acute myocardial infarction. You see here the thin apex, there's no muscle anymore. And of course, such a heart can no longer adequately pump, and this leads to heart failure. It's even worse in aging. Their heart function is already compromised at baseline. And further, there is an infarct expansion and after infarction, a worse outcome in the aging heart. It's also nicely introduced, besides cardiomyocytes, which are the cells, of course, which are contracting. And of course, if they are dying, this is difficult to compensate for. There are many other cells which control this process and provide paracrine factors which can either promote or prevent cardiomyocyte death. Cardiomyocytes can increase in size to compensate in part for cardiomyocyte loss, or they can also proliferate, as I said, but this is only seen in postnatal stages and very rarely occurs in adult hearts. And this coordinated fashion of the cells, which comprise of fibroblasts, which are responsible for generating matrix, immune cells, which also can be positive or negative regulators of cardiomyocyte death and growth, as well as the vasculature, which is of course also important in the heart to provide oxygen, but also provides paracrine factors. This all needs to function in a consorted manner, and this is what I'm going to talk about. I will present you two stories, one of which is the effect of aging on the heart. So without injury, what happens to a heart under aging? And this study was mainly driven by Julian Wagner. And a second study addressing the same question in the end, what happens in heart failure in humans? Why aging? As already alluded to, aging is one of the drivers of cardiovascular disease. It leads to an increase in the clotting of the blood, which then leads to myocardial infarction. And myocardial infarction leads to a phenotype called heart failure with reduced ejection fraction, meaning the heart can no longer pump. You see here an expanded heart, and this can no longer function properly. In addition, aging without affecting the vasculature can lead to heart failure with preserved ejection fraction. This is mainly related to diastolic dysfunction, meaning the heart can no longer expand, it becomes stiffer. We have a lot of matrix and fibrosis ongoing. and this leads also to dysfunction. And both of these types of heart failure are equally contributing to death of people and are equally also affecting the lifestyle, meaning you cannot walk anymore, you cannot respond to, for example, if you walk stairs, this is no longer possible if you have heart failure. So we addressed first how aging affects the muscle and the cardiac tissue. And we did so by comparing mice hearts. So we used 8-month-old mice compared to 12-week-old mice, and then used a new technology which mainly dissects the individual cells of the heart, because we felt that in the past, the focus was mainly on cardiomyocytes, but the cells around the cardiomyocytes may be of equal importance. And this single cell sequencing, for those of you who are not familiar with this means that you isolate each cell by a droplet-based approach, and you sequence each cell, and then you can define the transcriptome in each cell population instead of looking in the average gene expression of all cells, which of course is very difficult to get insights if you have heterogeneous composition as you have in the heart. So one of the students, Julian, when he did his PhD thesis, he said, this is like looking in a fruit salad instead of looking in the smoothie. And of course, you now can depict cardiomyocytes and all the other cells and their response to aging. This is the data, just to summarize how this looks like. This is now the bioinformatic analysis of the single cell sequencing data. You group them in the end by the neighborhood to similar cells, and then you see clusters of cells, for example, cardiomyocytes, endothelial cells, immune cells, fibroblasts, and so on. And this were now 6 hearts, 3 young hearts, 3 old hearts, and you can appreciate all of them contribute to the cell populations. And then we asked the first question, so what is affecting, or what is happening in aged hearts? This shows you here now the differentially expressed genes. In the upper ring, you see here the upregulated genes in a different cell population. In the lower ring, in the inner ring, you see here the downregulated genes, and the colors encode for the different cell population. And you can easily see the pink color depicting fibroblast population A together with the darker red here, fibroblast population B, comprise the majority of dysregulated genes. So aging has a major impact on the fibroblast population, followed by the cardiomyocyte population here in blue, then endothelial cells and other cells responding to aging. You know, aging is a general phenomenon, and we were questioning whether aging leads to a similar age-associated phenotype in all cells, but this was not the case. So this shows you now the differentially expressed genes in the different cell populations, so cardiomyocytes, endothelial cells, immune cells, and fibroblasts. And we were expecting that the majority of cells might be similarly regulated, like senescence-associated genes or something like this in all cells, but only 11 genes were regulated in all cell populations. The majority of cells was very cell type specific. For example, 126 genes being regulated in fibroblasts and 47 genes only in cardiomyocytes. This suggests that, first of all, the fibroblasts respond mostly to aging. I think this is not so surprising, but still we were surprised by the strong effect we have seen. And the second is that the genes are regulated in a cell type specific manner. To get some further insights in the cell population of fibroblasts, we subsequently now zoomed in the fibroblasts. So we only took the bioinformatic data from the fibroblast population and then used a tool called Pseudotime analysis with this monocle, where you can sort the cells, suggesting that possibly they may represent different states. This was used in development studies, but you can also use this if you compare disease, for example, to healthy. And you can nicely see that by using this approach, the cells are distinguished in populations associated with either age-associated states. These were the old heart-derived cells. So this is the red color. The blue color depicts young heart-derived cells. And you see the young heart states were higher here, and the old heart states were higher here. And of course, then you can question what is specific and unique to the age cells and what is specific and unique to the young cells. In the first cluster here, we found out that angiogenesis, which is a process referring to endothelial cell response to injury and vessel growth, was enriched. In the second gene cluster, we found inflammation, which is typically also seen in aging, being enriched in the 2-cell populations here in the aged hearts. And finally, interestingly, I will not speak about this, but what was really interesting is that fibroblasts acquired an osteoblast gene expression state, suggesting that they lose their identity. And trypsostase, usually you shouldn't see osteoblasts in the heart, but this can happen during aging. In young hearts, extracellular matrix structure and organization was high. And because we found that this cluster here being very significantly and strongly enriched in the heart, we then focused on the understanding of these angiogenesis-related genes in the diseased aged hearts. So we question whether fibroblasts, which show these changes in gene expression of angiogenic factors, might control or interact with endothelial cells, which are the cells usually responsible for angiogenesis, and therefore the Go terms in the fibroblasts may control endothelial cells. And we hypothesized that maybe in the aging heart there might be an impaired communication, as you see, as you also have it in humans, which can no longer listen and maybe also not talk adequately. Maybe also in the aging heart there might be a miscommunication happening in these situations. To get some insights whether cell communication might indeed be affected, we used again a bioinformatic tool. At that time, we teamed up with Ralph Patrick and Richard Harvey in Sydney, who at that time already had a very nice tool to address putative ligand interactions. And we did so by counting the expression of ligands, either being it membrane-bound or soluble ligands, and the expression of receptors. So if a cell has a receptor and a ligand, there could be an interaction, and this is counted as a potential predicted interaction. While if a cell type C has no receptor, of course, this cell type is not expected to talk to the cell type A. Of course, this is only now limited to the knowledge of ligands and receptors we are knowing, but at least there are a lot of interactions already known, and this is studied in this analysis. We then looked how cells could communicate, and it was interesting to note that in the aging heart, the majority of cells which are talking to other cells are fibroblasts. So the total outgoing weights, meaning the signals going out of the cells, came from fibroblasts, and the highest signal incoming in the aging heart were from endothelial cells, meaning that maybe potentially, and this was our hypothesis, fibroblasts indeed could speak to the endothelial cells by providing paracrine factors, and this could really change the properties and the endothelial cells, and may lead also to a change in endothelial cell function in aging. To address whether this hypothesis could be true, we then used in vitro culture studies to address the hallmarks of aging and looked into the endothelial response to the fibroblasts of the old heart. So what we actually did was to culture endothelial cells, and then at conditioned medium, which we obtained either from primary heart fibroblasts from young hearts or old hearts, used this conditioned medium and added this to matricial dishes, where we then can assess endothelial cell growth. You can compare here, these are control conditions. These are typical endothelial cell structures in control conditions. If we add young fibroblast mediums, nice structure formation. But you see here, if you add aged heart-derived fibroblast medium, you really see a disturbed structure of the endothelium, suggesting that fibroblasts from aged hearts somehow have a toxic or at least a harmful effect on endothelial cells. Additional hallmarks of aging, as you may know, is inflammation. And the inflammatory response of endothelial cells was also changed under the same experimental setups, not under baseline condition, but if you stress endothelial cells by stimulating them with TNF-alpha, you can appreciate further upregulation of a typical inflammation signature marker, interleukin-6. This is with young heart fibroblast medium. This is with old heart fibroblast medium. Again, a further aggravation of inflammation. Also, autophagy, interestingly, was significantly reduced. Autophagy is is an organelle reorganization and reuse, which is also important for maintaining tissue homeostasis. And endothelial cells showed a lower autophagy reaction when being treated with aged heart fibroblast medium. Interestingly, there was no direct induction of senescence, so proliferation of the endothelial cells was not affected. These data suggest that at least with medium of old heart-derived fibroblasts, we can induce a harmful effect on endothelial cells, reduced angiogenesis, autophagy, increased inflammation. And this, of course, might reduce endothelial cell function by maybe mediating a harmful crosstalk, amplifying endothelial injury. Of course, the question is which factors are responsible for this, and there are many factors I can tell you. There were many significant expressed genes. Among them were serpins. These are family members, serpin-E1 and E2, which were highly induced in the age-associated clusters. If you recall these fibroblast clusters, this one was the age-associated angiogenesis cluster, but also this one. There's an induction of serpin-E2. This was also seen with serpin-E1 and serpin-E2. And if you also look into serpin-E1 expression in isolated fibroblasts, there was a strong induction of protein expression in old heart-derived fibroblasts compared to young heart-derived fibroblasts. So the question whether the serpins might be responsible, at least in part, for mediating this impairment effect. And to address this question, we used neutralizing serpin antibodies to treat this conditioned medium from young or old heart fibroblasts, and then again use the tube formation assay to test the effect. And as you can see here, this is again the controls. These are the aged fibroblast mediums looking into an impaired angiogenesis activity. While if you add serpin antibodies, this is similar to the young fibroblast medium cultured cells, suggesting that these serpins, which are known to regulate angiogenesis signaling and also are important in homeostasis in vascular biology, that they may contribute to this impairment of endothelial cell function in the aging heart. Interestingly, serpins are also typical markers for senescent cells. And as you know, senescence is a typical feature of aging. So we question that maybe a therapeutic approach to prevent this harmful interaction might be by using senolytics, which can remove these senescent cells. And this is a study, it's still ongoing, but preliminary data in vitro suggests that indeed these senolytics might be protective. Senolytics are first described or are described in this study. You can use different types of senolytics, and quercetin here is one of the senolytics which targets the serpin-E pathway. And we use this to treat these young and old heart-derived fibroblasts, conditioned medium, the fibroblasts, and then took the conditioned medium and then assessed whether we can get rid of these harmful effects. And indeed, when we took these senolytics, we could in part rescue these functions. So this is again young fibroblast medium, this is old fibroblast medium, and this is the same when we treat the fibroblasts before with quercetin. So it appears that the senescence in these cells, in the fibroblasts, is somehow involved in the negative impact on these endothelial cells. And you see here a rescue, at least in the tube formation assay, by the treatment with senolytics. As said, we are now doing in vivo studies to further confirm these findings, but these in vitro studies bring first hints that senescence may play a role, senescence of fibroblasts may play a role in vascular dysfunction in the aging heart. With this, I would like to conclude the first part of the study and come to the second part. This is actually done by a very, very good team of researchers, Luca Nicin, who is currently finishing his PhD thesis, Simone Glaser, she is a postdoc, and Sam Schröter, who is a MD student, and they together wanted to understand how human heart failure is controlled. You may understand that getting human heart tissue is not so easy. So these single cell technologies really allow us to gain now first in detail insights into the human heart failure. And also as mentioned in the introduction, heart failure is a major reason for death. It can occur secondary to myocardial infarction, but also it can occur in response to aging and comorbidities leading to diastolic dysfunction, as I also showed you in the first slide. It can also occur in response to pressure overload and of course also genetic diseases. And the first study which we now completed in humans is a study where we investigated the effect of pressure overload. Pressure overload is typically occurring in patients with valve stenosis. You see here the valve, which is let's say it's responsible for keeping the blood outside the heart, but now then the heart needs to pump out the blood. And if you have a stenosis here, the heart can no longer pump the blood out of the heart, and this makes a pressure in the ventricle, leading to hypertrophy and then a growth of the cardiomyocytes and impairment of cardiomyocyte function. To gain some first insights, as said, we used the single cell technology, and here we used published data, so in part from human hearts, and pooled them with patients which we selected in our hospital from aortic stenosis patients. And this is really cool. So you can use published data now and also own data to combine them. And by combining this, you then can select the different cells of the heart. So you can see here cardiomyocytes, neural cells, endothelial cells, fibroblasts, and immune cells, and look in detail in each cell population and ask the question, what happens in humans with heart failure? I will not have time to share all of the data. This is a ton of information out of these data sets. I hope it will appear soon online, and you can look in more detail. But what was super striking in this study was that cell communication was mainly affected. I've introduced these cell communication bioinformatic tools, and now there are two more softwares available called CellPhoneDB and CellChatDB, both published, both available for everybody. And you can then appreciate the interactions of the different cell types in the heart. These are here cardiomyocytes. These are endothelial cells, fibroblasts, smooth muscle cells, and the other cells. And already by looking at these pictures, you see here that these red lines are much thicker in the healthy heart than compared in the hypertrophy heart. These are the lines indicating communication pathways between cardiomyocytes and these other populations. This is the same if you use another tool, CellChatDB, again, very strong communication between cardiomyocytes in healthy hearts and less so in hypertrophied hearts. When we sneaked into this data and looked in the number of cardiomyocyte incoming signals between the healthy heart and these diseased aortic stenosis hearts, you can appreciate that the signals were always higher in the healthy hearts compared to the diseased hearts. This was particularly true for endothelial cells, but also many of the other cells showed a reduction in incoming signals. Because of our interest in endothelial cells, we were a bit biased, I have to say, to begin with, and we studied in depth the communication pathways between endothelial cells and cardiomyocytes as a first step to gain insights here. These are now the top regulated genes. These are the top down-regulated cell communication pathways. On the upper panel, you see cardiomyocytes, and the lower panel endothelial cells. And you see here the communication paths. You see that there are many interesting molecules being regulated. VEGFs are very important vascular growth factors and their receptors. Semaphorins, angiopoietins, very important vascular stability factors. But what drew our attention, which has so far been not so much studied, were free Ephrin and receptor ligand interactions, which were disturbed in the aortic stenosis patients. Ephrins and Ephrin receptors are very important in development, cell differentiation, tissue, fibrosis, inflammation, and angiogenesis. And they come in many flavors, and there are many receptors. You see here, these are the Ephrin receptors known in humans. Here the Ephrin ligands, so these are the ones which activate the receptors, also several paths, and they can crosstalk to another. Interestingly, the receptors are expressed and are activated by the ligands, so the Ephrins are the ligands. But the ligand itself can also induce a reverse signaling, so it's in a way a crosstalk between cells. If they talk to each other, there's a feedback to both cells. And to get first hints in what could be relevant here, you see there are many receptors. We looked in which are the receptors which are highest in the cardiomyocytes. And you can appreciate here, this is EFNB1, this is Ephrin-A4. These are the highest expressed Ephrin receptor in the cardiomyocytes. And if you zoom in here, the Ephrin-B1 receptor, this is now each line represents one cell. Red means high expression. White means no expression. And you can appreciate in the healthy heart a very high expression in cardiomyocytes, in the stenotic heart a low expression. And this is a profound downregulation of Ephrin-B1, but also Ephrin-A4 and some other Ephrin receptors in cardiomyocytes in these diseased hearts. To sneak a bit in the and see whether this is indeed true, this is all, as I said, is bioinformatic prediction. Here are some raw data. So these are single cell sequencing data sets with violin plots, with single cell nuclear sequencing, I must say. You see here EFNB1 expression. This is a total of all cells, all nuclei which we have in a healthy heart. And these are the total of the aortic stenosis. And you see here only a few expressing EFNB1 in the healthy heart, a very nice expression. And this shows you now these are 14 healthy human hearts and 5 diseased hearts. And you can really see very homogeneous high EFNB1 in the healthy and low EFB1 in the diseased hearts. Of course, this is RNA. What happens on a protein level, you can also appreciate here in immunostainings. There is a EFNB1 expression in a non-hypertrophied heart, while this is significantly reduced in the hypertrophied conditions. This is a study where we get the samples for this validation by Benjamin Meder and Christoph Reich from Heidelberg to validate our RNA sequencing data. This is a mouse model again, because it's a bit more clean, and also here we found a downregulation of the Ephrin protein expression to confirm these data. What is now the relevance? The function of Ephb1 in cardiomyocytes is unknown, and we were wondering how this receptor can interact with other cells. And first of all, studied also the ligands. And one of the prominent ligands of EFNB1 is EFNB2. And interestingly, as I already alluded to initially, EFNB2 is very highly expressed in endothelial cells. So these are again these plots where you can see the different clusters of cells. This is cardiomyocytes, which express the receptor. And here the ligand is really strongly expressed by endothelial cells. You see also in the extracellular, in these plots here, that EFNB1 is mainly localized, predicted to localize in the membrane. And the ligand can indeed be expressed also by the membrane, but also can be in the extracellular space. So there might be a communication between endothelial cells and cardiomyocytes. To test whether this is indeed the case, and since Ephrin receptor B1 has not been studied in cardiomyocytes so far, we added recombinant EFNB2 and looked whether the phosphorylation of the receptor is active. And this is indeed the case. You see here an induction of phosphorylation by recombinant EFNB2, suggesting that there might be a crosstalk. But if you go one step back and think about how could these cells crosstalk, so we went also in electron microscope images, because usually there is also matrix in between cells in the heart. And you see here endothelial cells, these are cardiomyocytes. The image is kindly provided by Guillermo Luxan, and he looked into it. And usually you have extracellular matrix space between endothelial cells and cardiomyocytes. But if you look more deeply, you also find areas where cardiomyocytes interact directly with endothelial cells. So there indeed might be paracrine interactions between the cells, that like in this setting, but also there might be direct cell communication by direct membrane communication pathways, like a receptor ligand interaction could occur also in the heart. So the question was, could endothelial cells expressing EFNB2 interact with the Ephrin receptor B1 on cardiomyocytes, and could these maintain healthy cardiomyocyte? To gain first insights in vitro about the function of the EFNB2 and Ephrin receptor B1 interaction, we again used recombinant EFNB2 and assessed whether cardiomyocyte hypertrophy, which, as I said, is a response of the diseased heart, is affected. And indeed, you see here, if you stimulate hypertrophy by a substance called PE, phenylephrine, which induces hypertrophy, the recombinant EFNB1 prevented this induction of this response. You see it also in these images here, enlargement of the cells, which is prevented by the recombinant EFNB2 addition. Okay, you may say this is cell culture. So we also used, together with Chaya Krishnan's group, a human cardiac organoid model. This is a nice model where you have all the cells of the heart. By using iPS-derived cells, you can really generate multicellular organoids, and we looked into the response of these organoids. They also are contracting, so they are really viable and more closely resembling cardiac tissue. And again, you see an induction of hypertrophy by this phenylephrine stimulation, and again, an inhibition by adding recombinant EFNB2 to reduce the volume of the cardiomyocyte size. But recombinant Ephrin-B2 will be one way, but can endothelial cells really talk to cardiomyocytes directly by expressing the Ephrin-B2? To gain insights into the direct cell-cell communication, we silence Ephrin-B2 in endothelial cells and then co-culture them with cardiomyocytes. And to me, this came as a surprise. This is the culture effect. You see here the endothelial cells on the bottom, and then we added cardiomyocytes on the top. You see here the nice striation of the cardiomyocytes under control conditions. And interestingly, when we silenced Ephrin-B2 in endothelial cells and then added cardiomyocytes, the cardiomyocytes gained increase in size. So there is a hypertrophy induction, even just when removing Ephrin-B2 in this model. You can see the quantification on the right-hand side. There is an induction of cell size, cardiomyocyte hypertrophy. If you use endothelial cells which lack this ligand, also the contractility was reduced. So there was an impairment on function. And we could measure also cardiomyocyte stress markers by using rat-specific primers, because the cardiomyocytes were from rats and the endothelial cells from humans, and could measure stress responses, genes like NPPA, NPDB, ACTA1. These are all typical stress response genes, and all went up under these conditions. This suggests that the lack of Ephrin-B2 in endothelial cells is sufficient to induce cardiomyocyte stress. And we also, I will not have time to show you, but we rescued this effect, so suggesting that it's really the Ephrin-B2 in endothelial cells which is responsible for this pathway and for this response. So this would suggest that endothelial cells via this receptor interact, and the question was whether we can also rescue cardiomyocytes by augmenting the expression of Ephrin-B1. This would be a way for therapeutic development. So in the diseased heart, we could consider augmenting this receptor to restore function. And at least in vitro, we found nicely that overexpression of an AAV encoding Ephrin-B1 led to a reduction of this stress response, also again in these cardiomyocyte cell cultures. So we see again a stress response, hypertrophy induction by PE stimulation. This was prevented and reduced by overexpression of this receptor. This would suggest that maybe, and this is what we are currently testing, whether the receptor overexpression might be a therapeutic tool also to prevent heart failure, or at least to ameliorate further progression of heart failure. With this, I would like to conclude this study. We have seen that in heart failure induced by pressure overload, leading to hypertrophy, there is this change in the overall cell communication pathways in the heart. I've shown you the example that Ephrin-B2, Ephrin-B1 interactions are important, but there are also more in this data. I will not have time to go. There are really disturbed interactions also with fibroblast-derived factors and other signaling pathways, suggesting that not only aging, but also disease is associated with a really strong change in the communication pathways, which may lead to a reduction also of active and protective signals and an inactivation of these protective signals, which may contribute to heart failure. One of our colleagues, Katharina Schulenburg, and together with the students made this nice cartoon to bring this to a more general audience. So we felt that this means that under healthy conditions, there is a lot of talking between cardiomyocytes and endothelial cells among this Ephrin-B2, but in the diseased hearts, the cardiomyocytes, because losing these receptors, may no longer be able to listen to these environmental factors. And this may lead to an unhealthy phenotype. And we hope that by restoring the receptors, this might be making the cardiomyocyte healthy again. So this is just as maybe a teaser. We need now to understand the mechanisms of these effects and the downstream signaling. I mentioned that Ephrin-B1 has not been studied, so the signals, pathways, and maybe also pharmacological approaches might warrant further research. And if you want to follow our research, these are our Twitter accounts, so maybe you can sign up and see what's coming out in the next weeks and months and follow what we are doing. With this, I would like to thank the entire team. I have highlighted already some people, but it's really the entire team and many others who contributed to this endeavor, also collaborating partners, and just again highlighting Julian Wagner for his initial studies on aging, Simone and Luca for the together with Sam for the communication, And particularly, we couldn't have done this without David John, who is the head of the bioinformatics group, Ralf Schulz-Brüning, who was a student who helped us to combine the data sets, and Wesley Applamalt, who did all the wet lab stuff for the single nuclear isolation, and Lucas Tombo, who is really a wonderful bioinformatician as well, and also works in the wet lab and helped a lot for data analysis. And of course, our clinical team and the team of Chaya Krishnan for providing these nice organoids, as well as the hospital of Andreas Sayer and the surgical colleagues, and also our colleagues where we also study now age-dependent signatures in kids with heart failure. So just highlighting that the research goes beyond the study of adult heart failure, but we also look into children which have genetic diseases and have really a very poor life expectancy. With this, I would like to conclude. I'm happy to address any question. Hope that you got interested in heart research, and particularly also in this understanding of cellular communication pathways. Thank you.
Thank you, Stefanie. Thank you for a very interesting talk. It's really thrilling to see so many interactions between endothelial cells, fibroblasts, and endothelial and cardiomyocytes. So we have some questions now. So the first question is from Pascal Zimmerman. He wants to ask, did you look at PDZ proteins for the cues that might control Ephrin F communications?
This is a very interesting question. I think, as I mentioned at the end, we do not know yet the signals. These are really interesting molecules, and it could be very important to study this. We are currently doing proteomics and looking into actions and phosphorylation studies, but we have not yet looked into it. This is on the list, and the experiments are ongoing.
Okay.
I would assume that this could be very relevant, yes.
Okay, so the next question is transdifferentiation, that is change of fibroblast gene expression, that fibroblast triggers vascular or valvular calcification to lead to pathology like calcific aortic valve stenosis. Any thoughts or data on it? So, of course, we took the ventricle of the heart for our aged mice studies. But there it was very clear that we found some calcification signatures in fibroblasts. We also see them particularly in the epicardium. I think this study could be even more relevant if we look in the valves, which are calcified, and of course aging. So it could likely be relevant that there is really an osteogenic differentiation. We just downloaded some data from aortic valve stenosis patients and hope we can get some insights into humans to see whether these osteogenic signatures, which we have found in the aging mouse heart, may be also the same in the human heart. And I think this is a very interesting question. And like I would expect that there is something going on also with transdifferentiation of fibroblasts. Great. So the next question is from James Cranley. Thank you for the talk. You showed significantly different Ephb1 expression between diseased and healthy reference. This reference includes 6 regions with significant interregional differences, as well as several CM subtypes. How can you be sure that the difference in Ephb1 you see are not due to the fact that you sampled from different regions?
Very good question. This is an expert. So what we use here are location match samples. So we use samples from septum, in the end septum from the healthy heart and also from the diseased heart to match this. And also our validation studies, they were done from the other parts of the heart, but also location matched. So we are quite confident that it's not affected by the localization of the heart. But I have to say, not for this study, so it's not a confounding factor, but it's very interesting to see how different regions of the heart show different signatures. So also in this original Nature paper, which I cited, there are striking differences, and also in age charts, we see differences in the right versus left ventricle. So it's worth looking into more details and not thinking that the whole heart responds equally. So spatial transcriptomics will be important. There may be also gradients from epicardium to endocardium. And I think this deserves further studies. It's a very good question.
Yeah, thank you. while we have more questions to come in, so can I ask you something? So initially you showed this age and young heart and their single cell data. So certainly there is increase in fibroblasts with age. So can you shed more light on this, like how the fibroblasts are increasing with age? Are they coming from any other cells within the heart?
So it's developmentally, fibroblasts come from different regions, but in the aged hearts, I think all of the fibroblasts are expected to proliferate because it's known that fibroblast proliferation drives the increase in fibroblast numbers. I can't tell you exactly whether the different origin has the subpopulation, because they are quite similar. So without tracing, it's almost impossible to really define the different subtypes. But we also have seen some fibroblast subpopulations by bioinformatics. So maybe there are some subsets which are more senescent around the vessels. This is something we are currently looking into it. We have some senescent signals, not overall in the heart, but around the vasculature. So we speculate that maybe the fibroblasts around the vessels may be more senescent.
Okay.
We need to do double stainings and maybe spatial transcriptomics to find this out. I hope that's your question. So I can't tell for the specific origin of the fibroblasts which are driving the process, but would maybe assume that it's more like a second phenomenon, which is stress responses and maybe location response.
Okay, thank you. So there's a question from Tobias Weinberger. So in your opinion, what relevance do immune cells play in the aged heart? Are they protective or may they even enhance disease? Did aged heart show a pro-inflammatory gene profile in immune cells? Yeah, 3 questions.
We profiled immune cells, but we did not further work on this because it's a ton of data and there's a major change. So we see complement activation and a lot of inflammatory signals. I think your question is very good because inflammation can be good or bad. And depending on the extent and the duration, inflammation may be protective or harmful. We don't know exactly what happens in the aging heart. We see quite an induction of inflammation, which I would assume, if this is chronic, may be harmful. But there may be also protective signals within these populations, like tissue-resident macrophages and things like this, which we haven't looked in detail. So it's a super question. If you come to us, you can study it. I think we have tons of data to look into it, but we did not yet follow this.
Yes. Yeah, I think that also may be a connection how like aging, there's an increase in inflammatory signals and also with the disease incidences that may give more information. I agree. Yeah. Thank you. Don't have any more questions at the moment, so maybe I can drop in another question. So is this like from the single cell data? So how are you taking this work further? Like whether the next stage would be to targeting any particular targets to reduce this age-related changes, or what's your next step?
So it depends, but of course there are so many interesting questions that I really have a hard time to focus on the most interesting one. And of course we now want to follow the EFNB1 study because the results, in vitro results, in my view, are very interesting. It's not yet studied, and we want to develop maybe a therapeutics to either overexpress Ephb1, or maybe we find out also downstream signals which could be reactivated by small molecules or things like this. Therefore, I'm interested in the signaling also of EFNB1. I think with respect to aging, these senolytics would be interesting, but my dream is to have a more specific senolytics, to not, it's a bit like senolytics kill all senescent cells, and senescence is also a kind of important for repair stress responses. So I would like to know better about vascular senescence and fibroblast senescence, and maybe more specifically interfere with these pathways. And this is, of course, what single cell sequencing data also could be used to then look in specific senescence pathways, which might be typical for the heart and be more organ specific instead of just treating the whole body with something which is globally affecting. This is what, but there are many other interesting questions. I think inflammation is interesting. Senescence per se is interesting. I also think that now understanding the different types of heart failure, which we can do by combining these single cell sequencing, like heart failure, age-associated heart failure, ischemic-associated heart failure, and all of this, they learn much more about the tailored treatment of patients. Instead of giving all patients maybe similar drugs, maybe we get more insight into specific attempts for specific types of diseases.
Yes. Yeah.
This is it's a difficult question because we could about 2 hours and have to exchange ideas.
I know. Thanks. So there's another question coming. So do you think there will be difference in endothelial cells communicating with endocardial myocytes versus epicardial myocytes? From Priyanka.
I don't know. I didn't look into it. I just know that in aging, we see changes in endothelial cells, also spotty. It's not that there is the whole heart and they react similarly. We have to look in more details. It's a good question also.
Okay, so there are no more questions. So let's thank Stefanie for the wonderful talk and discussion. I think we can start now with a close meeting with the PhD students. If they have questions, they can just ask questions.